Hello, everyone. Thank you. Thank you very much for joining. My name is Ricardo. I'll be hosting today's webinar. So welcome to our first gold standard diagnostics webinar of the year, or GSD, as I will call it from now on for short. For this first webinar, as they say, we wanted to start with a bang. So we tried to pick a topic that would be of a widespread interest. And I hope we achieve that here because, at least for me, knowing what the food industry is doing about allergens, by that I mean what are the methods they are using, what is the industry tracking, new regulations coming up, what are the new allergens that are coming up, I think is of a widespread interest and a pertinent question. And we hope that these are some of the questions that we can answer today. However, when I say we, I actually mean she, because this is clearly a one-woman show, as Christina, which you'll see in the next slide, is the PM, for product manager for Allergens. She'll be the one presenting today and giving the ins and outs of the topic and answering your questions by the end. And my role here, you can see me over there too. I'm the product manager for pathogens at GSD2. But I'll be here as the hosting and moderator and asking your questions in the end for Christina to answer. Now, who is Christina, you might be asking? And I can tell you right away that she is quite humble and she would not want me to extend too much on this part. But I'm sorry, Christina, I still have the microphone, so I'm sorry. I can tell you right away that she's a joy to work with. We here at GSD, we have a culture of product managers to work very closely together. To learn as much as possible from each other and basically share experiences. And with that, I can really tell she's a joy to work with, and she really knows her stuff. So she has a biochemist background. She specialized in biotechnology. And what I keep noticing whenever we go to a congress or a meeting is that everyone in the industry seems to recognize her. At least one person will come to us and will know Christina. And that will not be a surprise when I tell you that she has 15 years of experience, first on the food safety line, and then more recently as a product manager. So yes, she really knows her stuff and she will definitely be able to answer your questions today. And this is Christina then. Just before moving to the actual presentation, just a quick overview on who we are. So as I mentioned, first, we are gold standard diagnostics. We are a fast-growing global provider of diagnostic test kits, lab consumables, ELISA-based instrument platforms that support this rapid testing in food, feed, environmental, animal health solutions, clinical diagnostics. So if you have any kind of questions or doubts or want to know more about any of these topics, please feel free to reach out to us and we'll be glad to answer those questions or get back to you as soon as possible. And I promise this is the last thing in the next slide. Just some housekeeping topics here. If you have any questions, please use your GoToWebinar panel. You can see over there, usually on the right, you can see the question tab. You can answer your questions. You can ask your questions there, and we'll be sure to answer those questions at the end. And of course, if we don't have time to go through all the questions, we'll reach out to you afterwards, and you can feel free to reach out to us at contact@eu.goldstandarddiagnostics.com. And without further ado, I hope you do enjoy this presentation. And without further ado, as I said, Christina, the floor is yours. Thank you very much.
Thank you very much, Ricardo, and thank you very much to all of you for attending this webinar. So, okay, as you can see in the agenda, we first are going to see some short introduction about the food allergies. Then we will talk a little bit about one of the hot topics right now in the allergen field, which is the new and emerging allergens in the food industry. Then we will see some of the new challenges that is facing the allergen legislation and different techniques that are currently available for food allergen detection. And of course, we will show you which are the GSD solutions able to cover the entire allergen management process. And at the end of the presentation, as Ricardo has mentioned, we will leave some time for you to ask some questions. So I hope that we will be able to answer as good as possible. Okay, so let's start for the introduction. And I always like to start with this slide because I think it's very important here in this area, in this field, to know the difference between food allergy and a celiac disease. So a food allergy is an abnormal response of the body immune system when intaking some kind of foodstuffs. So this allergic reaction may be mild, but in some cases it can cause severe reaction, even can lead to death. So we are talking something about really, really serious. On the other hand, we have the celiac disease. This is an autoimmune disease that happens when the ingestion of gluten leads to damage in the small intestine. So maybe it's not so severe in a first stage, but if the celiac person goes on taking gluten for a long time, finally it can lead to very important damage, like for example, even intestine cancer. So I think this is important role here, what we're talking about in this webinar. It is estimated that celiac disease affects 100 people worldwide, and approximately between 200 and 500 of adults and 4 and 10% of children are affected by food allergies today. So as you can see, we are talking about high percentage values. Currently, the only treatment for food allergies or celiac disease is to maintain a lifelong adherence to a strict allergen-free diet. Usually, food allergens are present as an ingredient, so they are listed in the labeling of the product, but sometimes they may be also be present inadvertently via cross-contamination, for example, when sharing food processing machines. So this implies a traceability of the food production process from the raw material to the final product. So it means that the food industry needs to perform an identification of food ingredients, components, and additives, and also monitor any potential cross-contamination. And of course, the most effective way to perform both activities is by an allergen analysis. This is currently the European regulation for allergen labeling, and it includes 14 allergens that are listed and should be labeled in any kind of packaged foodstuff. So we have here from cereals containing gluten, crustaceans in egg, until lupin and sulfites. But this is the European regulation. So we will see here that it needs to face some challenges, and well, we will see it more in detail later. But first, let's talk a little bit about the new and emerging food allergens. As we have just seen, the European regulation mentioned 14 allergens that need to be labeled in all kinds of packaged foodstuff. But there are important and severe allergic reactions to many other types of food. So since this regulation was approved, and we're talking about 2011, additional food allergens have emerged, playing an important role to be considered as part of the industry allergen management. So here I put some examples, probably one of the most important. So we're talking about fruits and vegetables, which may cause what is called the PFAS, which means the pollen food allergy syndrome, and it is caused by cross-reacting allergens found in both pollen and also raw fruits, vegetables, and some tree nuts. So it is affected by both kind of food and also pollen. We have here also the legumes that are not included in the European regulation, and mainly peas. Peas, because pea protein is used as an alternative for animal protein, and it is even more dangerous as an allergen that when we use it as raw material, because of course it has more protein content concentrated. We also have the buckwheat. So as you probably know, buckwheat is one of the cereals that is widely used as an alternative for the celiac people because it's a safely gluten-free product. So it is widely used for mainly for bakery. But it is true that more and more there are more people that is allergic to buckwheat. So in fact, in some of the some not European regulation, buckwheat is included like one of the allergens that should be listed in the ingredients of the food. And probably one of the more new ones, or one of the more modern ones, is the edible insect. So right now, there are 4 types of insects that are approved as ingredient to be used as a source of proteins. So it means that usually people that are allergic to mollusk of crustacean, which is they are in fact needs to be labeled as allergic. Usually these people are also allergic to the edible insect because of the kind of protein covering the insect is very similar to the mollusk and crustacean. So all these new and emerging food allergens should be taken into consideration for the well, to have a look at the current legislation. So which are the challenges that this allergen legislation is currently facing? First of all, difference by countries. So we have seen the European one, but when you go to a different country outside Europe, you can find that the legislation is completely different. For example, in Japan, buckwheat, oat, so they include completely different, some other different allergens. So this is very important problem for the allergic people when traveling, because the kind of label that you are going to find in different countries is going to be completely different than what you are used to in your own country. And okay, this is something also important to consideration. Also, all other challenge is the allergen management on site. So an effective allergen management always covers all aspects of the food product supply chain, from sourcing raw material through manufacturing and packaging to the finished product that is sold to another business or to the end consumer. And for being able to implement an effective AMP, it's necessary to apply a documented systematic approach to identify and control all kinds of allergens that could be in place. And here in this allergen management, there are 2 key points, very, very important, which are the cleaning and the allergen risk review. When we talk about cleaning, we mean, or it includes all the procedures to manage raw material spills, cleaning the facility, the equipment, and all the tools to prevent allergen cross-contact that could be in place. And when we mention about allergen risk review, this is the throughout investigation of the allergen status of a food product. So it identifies the presence of allergens that are intentionally formulated into a product, so they are part of the product as an ingredient, but also quantifies the risk of allergens which may be unintentionally present. So it appears because of cross-contact allergens or cross-contact with other allergens. The third challenge, of course, is the limits, the regulation of the limits. So currently, only there are regulated limits for gluten. According to the Codex Alimentarius, is if a product contains less than 20 ppm of gluten, it can be labeled as gluten-free. If it contains between 20 and 100 ppm, it can be labeled as low gluten. And if it contains above 100 ppm, so it should be labeled as containing gluten. So in fact, gluten should be included like one of the ingredients. It is also important here to consider that the reference method for gluten determination is the R5 method, which of course is the one we have available. But this regulation or these limits have been only established for gluten. For the rest of the allergens, there are no regulated limits. So usually when we develop a new kit, what we always look is for the lowest limit of detection as possible, the highest sensitivity, because in order to avoid as much risk as possible. And the last challenge, but not less important, is the peculiarities about allergen diagnostic. So in all the cases, we are talking about complex commodities, protein mixtures. Many times these commodities have been heated, hydrolyzed, fermented. So here you have an example only for soy. When you go to buy soy in the supermarket, you can find it as unroasted soybean, roasted soybean, tofu, texture, granulated soy, soy milk, soy sauce. So many different formats. And when all these kind of formats, all the kind of matrices may have a different behavior when doing an analysis. And this is not considered in the legislation. It should be also be considered. For one other of the example is some heat-treated samples like egg white usually provides lower recovery that when the sample has not been heated. And of course, the cross-reactivity between biological variants. One of the most clear example is the mustard. So here you have a table, and of course, it's quite logical that the kits cross-react with different kind of mustards. But here you can also see that there is a high percentage of cross-reactivity with rapeseed, which of course is not mustard, but it belongs to the biological family. So these are some of the challenges that we are still facing when doing this kind of analysis and when writing the legislation. Okay, so let's go now to see which are the different techniques that are currently available to be able to detect allergens in food. So, of course, we are going to start with the immunoassays and for sure the ELISA kit or the ELISA technique, because this is a reference method and because it detects the protein, and the protein is the one that provokes or causes the allergic reaction in the patient. So ELISA means enzyme-linked immunosorbent assay. It is based on an antibody-antigen reaction, and precisely this reaction provides the specificity, sensitivity, and also multi-matrix characteristic. And this multi-matrix is very, very important. As we have just seen, we are dealing with many different kind of matrices, different treatment. So it's very, very important that this kind of flexibility when doing the analysis. So here now I'm going to explain you the technical basis of the ELISA sandwich, because most of the kits we have in the market for allergen detection are sandwich one. There are also other types, but mainly most of them are sandwich. So we have a plate with small wells, and these small wells are coated with antibodies. Then we add the standards and the samples and incubate. If the sample is positive, then the antigen, I mean the protein, the allergic protein, will bind to the antibodies which are coating the plate. Then we wash to eliminate all non-specific binding, and then we add the conjugate. And what is the conjugate? The conjugate is usually the same antibody which is coating the plate, but it has an enzyme binding on it. And this enzyme is the one that later, at the end of the assay, it's going to provide color in case the sample is positive. We incubate, and during this incubation, you see that this structure is formed. So we have antibody, antigen, or protein, and then antibody again. So it's like a sandwich. This is why this ELISA kit is called ELISA sandwich or double antibody ELISA. We wash again to eliminate all the excess of conjugate, and then we add the substrate of the enzyme and the stop solution. So finally, if the sample is positive, what we will see is a yellow color. And here there is an important point, because the intensity of this yellow color will be proportional to the amount of protein or the amount of allergen that we have in the sample. So it means that this technique led us to perform a quantification of the amount of allergen that we have in the sample. Okay, so the second technique for allergen detection is the LFD, the lateral flow device, that you probably all know because they are the one that we have been using during the COVID. And so we have a strip, and then what we need to do is just to insert the strip in the extracted sample, and then the sample runs through the strip in a way that if the sample is positive, we will finally see two lines. If the sample is negative, we will only see the control line. And in any case that there is no control line in the strip, it means that the analysis is not valid and we need to repeat it because something has happened, that the sample has not run correctly through the strip or whatever. So if there is no line in the control line, we need to repeat the assay. And the third technique is also one that at least you must know because of the name, the PCR, the polymerase chain reaction. And what we do here is just to exponentially increase the number of target DNA. So in this case, we what we do is just to detect the corresponding DNA to the protein, to the allergic protein that we want to detect. And if it is present in the sample, we do many copies of it in different cycles until we have a very high amount of molecules and we are able to detect it by different techniques, detection techniques that, okay, it makes no sense to mention right now. But this is the basis of the technology. So these are the three techniques for allergen detection. So now we are going to see which is the full solution that we can offer you from GSD. So in Gold Standard Diagnostic, we have here the whole list of different solutions we can offer to you. So first, of course, we have the ELISA portfolio, including the Gluten R5 reference method. Of course, different readers to be able to interpret the results of the ELISA, and also the possibility of an automation. We also have an LFD portfolio, which can be used as a qualitative kit, but can be also used as a quantification technique by means of rapid scan reader that we will explain later with more in detail, because I think it's quite interesting and unique solution we will find here. We also have the swabbing kit for taking surface samples, spiking solution, and allergen control material for the internal quality control of the lab or the factory, a matrix validation department, and of course, and coming very soon, is the new portfolio of PCR technique for allergen detection. So let's start for the beginning and for the SENSISpec ELISA product line. So here you have a complete list of the ELISA kits we have currently available in our portfolio. You will see that it is very comprehensive one, and we have all the we have ELISA kit for all the kits that are included in the European regulation S13442 for the celery, because there are no immunoassay technique, no ELISA, no LFD able to detect with a high quality the celery in food samples, and also for sulfites, because in the case of sulfites, they are not proteins, they are different molecules, so they need to be detected by a different technique, not with an ELISA. It's not possible. So here I would like to stop a little bit in the Gluten R5 solution we have currently available. We have 3 different kind of R5 gluten kits. As I told you, this is the reference method for gluten detection. We have the kit that we can call as the normal one, the quick kit, sorry, for with shorter incubation times, and the hydrolyzed gluten R5. The last one has been designed for detecting gluten in some specific samples where the protein has been hydrolyzed. We are talking about beer, whiskey, some syrups, some baby food that usually are partially hydrolyzed to be more digestible by the kids, these kind of samples. But I would like to focus on the first one, the SENSISpec Incathin Gluten R5, because this kit has been granted as AOSC PTM status for use with gluten-free bread, oat flour, and stainless steel surfaces. And probably most of you know how important it is to have this AOSC certification. Usually for the accreditation bodies, usually requires it for the laboratories or even for the factories, or at least it makes your life easier when you have this kind of certification. So this kit has a high sensitivity, 3 ppm of gluten. So as you can see, it's quite below the 20 ppm we were talking, or which is legislated by the Codex Alimentarius. And this kit is the one necessary for the ISO 17025 accreditation of the lab. And one important topic here, the kit does not include toxic reagent, even for the extraction. So you can do the extraction step of the gluten with no need of use any kind of fume hood. And in addition, the extraction buffer, in addition of not being toxic, is also included in the kit. So no need to buy it separately. You have already a safe extraction buffer included in the kit with the rest of the reagents. So we can say that we have the biggest product range in the market for allergen ELISAs with high sensitivity, accuracy, and reproducibility. Is easy performance. In most of the kits, it is 20 plus 20 plus 20 minutes. So it's not long time, it's not very short time. So you can do a adequate amount of sample at the same time. It's very easy to remember and all the same. So this is very convenient for the technician in the case that they are doing several ELISA at the same time, which is something that happens very frequently. We have a common extraction procedure for most of the kit. This is also very convenient because it means that you can use the same extract for different ELISAs. No need to do several extraction of the same samples. We have the kits available in a 48 and 96 well format, and we participate in different ring trials and PTs, FAPAs, ELA, BPA, and so on. We have the possibility of the automation. We will explain it later more in detail. And of course, as mentioned, we are currently working in new developments, mainly focused on the emerging allergen we have just mentioned. So we expect that along this year, 2023, we will have available kits for the detection of bee, insect, oats, or buckwheat and buckwheat. And for the reading of the results, I like the offer we have right now. To be honest, I really like this reader because, as you can see, it's nice, it's very compact, it's easy to use, it's intuitive, it's of course cost effective, and it's compatible to all the plates that are used in gold standard diagnostic, and even for some other competitors' choice. So it includes by default different kind of filters. But here for allergens, we read all the time in yellow color, so you only need the 450 nanometers. But it's true that some customers always like to use a reference wavelength of 620 nanometers, which is okay, but not strictly necessary. And of course, the automation. So up to now, we have been talking about how to do the ELISA in a manual way. So if you do it in a manual way, you need, for example, you may need an incubator, or at least you may need to take care about the temperature in the laboratory. You need a washer, or at least a squish bottle, or a multi-channel pipette to do the washing. And of course, you need a reader. But here we can offer a fully automated system. So here you only need to load all the samples and all the reagents and start the program, close the instrument, and just let it working until it provides you a full report with all the calculation already done and in a PDF format. So we currently have two different instruments, the Bolt and the Thunderbolt. And the main difference is that with the Bolt, you can do one plate, and with the Thunderbolt, you can do 2 plates. So this instrument includes an onboard camera, so it's very convenient because this way, even if the instrument is closed, you're able to see everything that is happening inside the instrument. It includes also a high precision syringe, and of course, it is validated. We offer them, or we provide it with validated protocols for allergens, but not only for allergens, also for mycotoxin, pathogens, water ELISA kits that we have available in GSD. And now we would like to see some of the complementary products to the ELISA kits, which, even being complementary, is very, very useful and very convenient for the customers. For example, the SENSISpec, the spiking solution. They are high concentrated standard solution for fortifying samples, and they have been completely validated with all the SENSISpec ELISA product line. So they're very useful for the customers if they want to have a sample with, the customers may decide, I want to have a sample with 20 ppm, 10 ppm, 30 ppm of whatever the allergens. So they are usually applied for process validation, matrix validation. This is very important because then you can see if there is some kind of background of some specific matrix with this allergen, and in general for continuous quality assessment. This is the spiking solution. We also have a swabbing kit for surface samples. The kit has been validated both for the ELISA and LFDs of our portfolio, and it can be used for wet or dry surfaces. You can also use it for in-house testing and also to take the sample and send it to a service lab. And we also have what we call the allergen control material. This is not the same as the spiking solution. This is something different. In this case, we are talking about an unprocessed material which has been already spiked with 2 times the LOQ of some allergen concentration. So we currently have 4 different kinds of allergen materials. The first one, we have called it key allergens because, of course, it contains the most important allergens: gluten, milk, soy, egg white. The second one includes crustacean fish and mollusks. The third one includes all kinds of nuts. And the fourth one includes all the sheets. So in this case, this is not liquid, this is a powder. So the customers should use it as a sample. So you need just to extract it and analyze it with the ELISA as a normal sample. And we provide with it a QC sheet, including some branch of values that this is the one that the customer should get. So this is also valid to know if the performance of the kit and the procedure you are using is being done correctly as an internal quality control. So now we have seen everything related with ELISA, and we are going to start with the LFD portfolio. Here I have included you the complete list of the LFD kits we have currently available. You can see that this is also a very comprehensive one, as big as the ELISA kits. And I would like to focus a little bit on the sensitivity of the kits, mainly in the case of surfaces. You will see that the values, the LOD values, are very, very, very low. So this is quite important because it's quite sensitive compared not only with the competitors, but also it's very low values compared with the ELISA technique. Usually, as you probably know, the LFD are usually less sensitive, but in this case, we have been able to get a very good sensitivity with this kind of kits. Here, for you just to see how simple is the procedure with the LFDs. So first, you need to do just the sample preparation. So it's just the homogenization of the sample and then the extraction. Then you transfer it to the incubation vial, and after 3 minutes, you can place the strip into the vial. 5 minutes later, you can then evaluate the strip. And as we have mentioned, if you see 2 lines, it means positive test. If you see only 1 line, it means a negative test. And only in the case of gluten, we have an additional dilution step, because in the case of gluten, the gluten proteins are not soluble in water-based buffer. So we need to use ethanol to solubilize them and to extract them. And then we need to dilute this ethanol so it does not affect the antibodies and the reagents that are included into the strip. But in this case, we are talking about a visual or a qualitative evaluation of the strip. But here, we can also offer you the possibility of doing a quantitative evaluation of the strip by using the RapidScan reader. So this reader has been specifically designed for field and in-process testing application. So it's not a big instrument, it's quite small. So it's portable, so it has a battery between 3 and 4 hours, so you can take it and move it to any place in the factory or in the lab. It's flexible, so only change the adapter, which is the black part in the lower part of the reader. So you can use it for different applications. You can use it not only for allergen, but also for GMO, water analysis, glyphosate, and so on. And probably the most important, it provides a number. So in some cases, this is very, very important. For example, for gluten. For gluten, we have a regulated limit of detection of 20 ppm. So it's not the same to get a value of 18 ppm of gluten that, for example, below 4 or below the LOD of the kit. So this is a unique solution in the market and very useful to get in a very easy way quantitative result for this kind of rapid tests, which are the LFDs. So as a summary, we can say that we have the most comprehensive LFD portfolio in the market, equivalent to the ELISA one. The kits are provided with all the necessary material to perform 20 individual analyses per kit. So we provide all the disposable material. We provide the pipettes, the vials, the spatulas, the swabs, even the swabs to take the surface samples. All is already included into the kit. You don't need any kind of additional material. We have a common extraction procedure for all the kits except for gluten. But as mentioned, we need ethanol in this case. But this is very, very convenient because with all the SENSIStrip kits, you can use the same extract to be applied in all the strips for all the allergens. They are easy to use lateral flow format. They give a rapid and accurate on-site test. And they have high sensitivity and specificity thanks to the antibody combination they have into the strip. And of course, very, very important, the possibility of quantification with the rapid scan reader. This is really a unique solution in the market. You probably will see readers, LFD readers for mycotoxin and for other kind of analytes, but not for all the allergens that as we have right now. So now let's go to explain a little bit about the matrix validation department. So the scope of this department includes two objectives. First one is to provide support to clients for their specific and problematic matrices. As you can imagine, when we develop a new kit or when we create a new kit, we try to test it with all kinds of matrices or with the most representative one that we can find in the market. But it's not possible to validate with all existing matrices. So in case that some customers want to really check the applicability of the kits to this specific or complicated matrix, we offer the possibility to calculate recovery, cross-reactivity, sensitivity, linearity, or maybe all together or different measure. Okay, we can adapt this validation report to the needs of the customer. The second objective is to perform a complete validation of some complex matrices, but not because of requests of a specific customers, but based on the needs we see from the market or the literature research we have done. So the general idea is to have an integrated solution to support the allergen testing process according with the customer needs. And of course, last but not least, the upcoming PCR portfolio. So we have talked about ELISAs, we have talked about LFDs, but what about the third technique we have mentioned? Of course, the ELISA procedure is the reference method. It detects the protein and we will go on being the reference method, but it has some gaps. So in some cases, like we have mentioned, for example, celery, there is no ELISA kit for celery detection. In some other cases, we can find that there is some kind of cross-reactivity. And in some other cases, we just want to confirm some not completely sure result with the ELISA kit or with the LFDs. So this is why we have, are working in the development of this new portfolio to complete the gaps of the current allergy portfolio we have available. So we have already selected, as a first stage, we have already selected these key parameters, the 10 key parameters. So please, I would like to call your attention to stay tuned because we expect to come with very good news in a short future about this new and very useful portfolio to really have a complete solution about allergens in GSD. So just as a general conclusion, I would like to say that an adequate control and allergen management is one of the current challenges of the food industry. And here in GSD, we offer all possible solutions and support to approach this challenge from different angles in order to assure the reliability of the industry and the safety of the end consumer, which at the end is the key point and the most important point. So with this, I would like to thank you, all of you, for your attention. You can see here in this last slide, so all the solutions that we have in GSD to cover all the food production channel from farm to fork, so from planting to the finished product. So thank you very much.
Again, that was a great presentation. Thank you very much. Thank you very much for joining. And we wish you all a great day. And until the next time, take care. Thank you very much.
Okay, thank you very much, Ricardo. Thank you very much to all of you. Have a nice day. Bye-bye.